So, welcome everyone, and thank you for attending today's webinar, Engage with AIBL, Data Discovery and Collaboration for EMCR. So, firstly, I'd like to acknowledge the traditional owner of the lands we are joining from today. So, I'm joining from Brisbane, so the lands of the Turrbal and Yugur peoples, and I pay my respects to elders past and present. So for today's webinar, we will begin with an overview of the AIBL study from our co-host, Dr. Tanya Porter, and then move into our three featured talks, which showcase a range of research made possible through the AIBL study. And then we will conclude with a Q&A session where you are welcome to ask questions about the AIBL study or directly to our speakers, or you are welcome to leave questions in the chat during the presentations. So now I will hand over to Tanya.
Thanks, Ying, and thank you to everybody for coming. We hope that this is will give you some information if you didn't already have it, or like some further clarification. But as a bit of a background to Ying and myself, so Ying we're both part of the AIBL, or we're both EMCR members of the AIBL SAC, I think of the advisory committee, scientific advisory committee. Ying is based at CSIRO in Brisbane, as she said, and I'm based at ECU in Perth. And our focus ranges. Ying is more in the neuroimaging side, and I'm in the genetics side. But essentially, just briefly, I would assume most people know at least a little bit of AIBL or have seen an AIBL presentation here and there. But the overall, I guess, aim of AIBL was to understand aging and the aging process, particularly Alzheimer's and how that progresses. So AIBL, I should say, stands obviously for the Australian Imaging, Biomarker and Lifestyle Study of Aging. So the broad aims include identifying biomarkers of AD, and that can range from imaging biomarkers to fluid biomarkers, whether that be blood or CSF, and then genetics. One of the things that AIBL has been successful in doing is tracking these long-term changes in the disease process, whether that be through cognition or cognitive measures or imaging measures, blood measures, and they're trying to characterize how we're changing from healthy aging to the different stages of AD, so MCI to dementia. One of the big parts of AIBL, which makes it quite unique with the addition of all the additional data, is how it aims to understand the impacts of lifestyle and health, and then also some of the genetic factors, and how these factors change your risk or resilience to the disease. AIBL has been, I guess, really big in the early diagnosis and early identification of some of these risk factors. And one of the big drivers for AIBL and what has kept us successful over time is, I guess, how good the data is, and that data has been used by researchers and industry partners to try and understand or work to improve testing and also therapeutics. So these are just a couple of, I guess, the big picture things where AIBL has been really successful. So one of the big things that AIBL has shown is that amyloid accumulation starts 20 to 30 years before the onset of dementia. And so this was work that was pioneered by people like Professor Victor Villamagna and Professor Chris Rowe. People like Professor Yang Ying Lim, identified how early cognitive changes are linked to preclinical amyloid positivity, and how that has worked to shape preclinical AD diagnosis. AIBL has been pretty big in the, big is the wrong word, it has published a lot of papers, peer-reviewed papers, worked towards the early development and validation of the centiloid scaling, and just contributed a lot of data internationally and nationally. So AIBL itself has two data collection sites between Perth and Melbourne, but the AIBL research team is located all over Australia. So again, big sites being in Perth and Melbourne, but also Brisbane, Sydney, and South Australia. And in terms of this, so Austin, the Florey, Nari, ECU, and CSIRO are the main management institutions, and they manage the study. But there are numerous other institutions that provide services, provide infrastructure, and work to keep the AIBL study going. So going through, I guess, what is available within AIBL. AIBL started in 2006. That's when recruitment began, and it began with around 1,100 participants being recruited as part of the inception cohort. And this included people who were diagnosed with Alzheimer's disease, had a level of mild cognitive impairment, or were cognitively normal or unimpaired. And the figure on the right is a little bit complicated, but it was from the AIBL paper that came out a few years ago that looked at 15 years of AIBL. And it just shows at baseline all of the participants that were in at baseline, and then how they have changed and developed over the course of the study. So in 2009, we started the recruitment of the enrichment cohort, and that is still ongoing till today. But essentially, participants were recruited to compensate for attrition, which there was a level of, expand high-risk groups, so MCI and LEAD, and also support new research questions. So additional data was decided it would be collected at this point. So in terms of what happens now, so there's still 18-month follow-ups, and this is across cognition, lifestyle questionnaires, and MRI, PET, and biofluids. Things have changed over time as new technologies have come out. So obviously with the introduction of tau PET in 2013, AIBL participants started having that measured as well. In the last few years, where there's been this uptake in blood biomarkers, and there's been additional capability to do that, there's been measurement of that in the cohort. I guess something that's close to my heart is lots of genetics has been collected over time. So we have where it started with only ApoE genotyping, that's been expanded to genome-wide genetic variant measurements, DNA methylation, DNA sequencing, and more recently we have gene expression data that's been collected. But all of this is to say that there's nearly 2 decades of longitudinal data. So for some participants, we have up to 10 repeated visits per participant where we're collecting all of this data. So to just go through a little bit about the actual data that's available within AIBL. So we have our fluid biomarker stream, and this includes measurements of blood and CSF biomarkers, things like amyloid beta, tau, NFL, GFAP, and other more specific markers, whether they have been collected on panels or for specific projects. We have neuroimaging data, so repeated measures of amyloid amyloid PET and tau PET, and then MRI measures of things like brain atrophy, so measurements of global brain volume, hippocampal volume, white matter, lesions, microbleeds, and more recently they have started collecting data on another PET marker, so the MAO-B marker for astrogliosis. In terms of the clinical and cognitive data, and this would be where the vast majority, well, the I guess cleanest data set, the largest data set within AIBL is the clinical and cognitive data. So there's very comprehensive cognitive and clinical cognitive testing across a large battery of cognitive tests and clinical staging as well. We also have things like mood scales, measures of depression, anxiety, and medical history. In terms of genetics, as I said, there's obviously ApoE genotype, which is, as everybody would know, the largest risk factor, genetic risk factor for AD. And then we also have those larger scale genome-wide variant arrays and sequencing. And lastly, we have highly phenotyped lifestyle information. So this includes questionnaires about diet, physical activity, and sleep. But there's also subsets where individuals have had objective measures taken, whether that be from accelerometers or, for example, DEXA imaging. So that is, I guess, in the nutshell, the data that AIBL has, and why we're encouraging people to incorporate AIBL data in projects is that it is a study that has this vast amount of longitudinal follow-up, so up to 15 years, with repeated assessments across a number of different data streams. So that includes, as I said, all of the measures of imaging, fluid, cognition, genetics, and lifestyle. It is a multi-site studies, but across those two sites where data is collected, there's standardized methods of collection, so between Perth and Melbourne. And I guess along the same lines of that, there are harmonized protocols across imaging. So no matter the site of imaging, there are harmonized protocols, the same as how the blood and fluid specimens are collected. The cognition is all collected in that way, and the same with the lifestyle data. And the data within each of these streams goes through very rigorous quality control and harmonization. In addition to within the study, everything being harmonized and controlled, there have been projects where AIBL data has been harmonized with international cohorts. So you can compare between those cohorts. So for example, ADNI, that started around the same time as AIBL, there are lots of data that is available that matches what has been collected in ADNI. And something that I think is also a really big positive for using AIBL is there's a strong collaborative network amongst the scientists, particularly I'd like to think EMCRs, and you'll get a look at some of the EMCR projects from Eleanor, Azadeh, and Emily. But yeah, there's a strong network of support amongst the AIBL scientists, and everybody is very happy to chip in and help with using the data. So I guess these are suggestions for how AIBL data could be used in your projects if it's something that you're interested in and something we're sort of encouraging. But if, say, for your project, you have a cross-sectional association with a study that you've developed, if you want additional longitudinal evaluation of that association, AIBL data is perfect for that. Just straight validation of an association you saw between, for example, a protein biomarker, an imaging biomarker, and something else. Validation of that. Something that, again, close to my heart is translation into the laboratory, but I guess back translation in this, forward translation in this way. I'm interested in the opposite way, but if there is a protein or a gene that you're seeing an effect of in a cellular model and an animal model, looking at whether variants within that gene or the expression of that gene or methylation of that gene has associations with some of these clinical phenotypes, I think it would add. Pilot data for grants. So if there are studies that you're wanting to develop or cohorts that you're wanting to develop, but you want a small amount of pilot data to show why that might be important, I think AIBL is a good place to start. And then also if you're in the area of statistical modeling or large scale sort of modeling, AIBL is a good place to do that. And there is so on Loney, which is a place where you can get publicly available data, there is a Loney data set that is not the full AIBL data set, but is very good for testing and training models that people often use. So I guess this is getting to the end of what I wanted to talk about today. But if you want more specifics on all of the data that I've outlined, there is a study paper that came out in 2021 that, as I said, was, I guess, an overview of the 15 years of AIBL and then also the AIBL website there. I think we might be sharing these slides, Ying, so yeah, I can send this around. But the last thing I wanted to talk about is, so if you are interested in looking into whether you could use AIBL or you have something in mind, on the AIBL website, there are expression of interest forms that you can fill out. So these are just a few tips and tricks I think is important if you want your EOI to be successful. The things that you're going to need are your details, obviously, a project outline. And so for the project outline, it's really important to clearly state the aims, hypotheses, and scope of what you're doing and be quite specific about what you're planning on doing and why AIBL is suitable. Then I guess the most important part is the data and samples required. So in terms of the EOIs that we've sort of all seen, the ones that are most successful and don't require back and forth typically are ones that specify exact data sets. So the measures that you want, not just saying all imaging, saying you want PET positivity versus negativity or measures of hippocampal volume, specific biomarkers, cognition, genetic variants and covariates. AIBL is always very supportive. Typically, potentially, if it's something that you want that's really new, the only reason sometimes that those don't get accepted is that if it's something that's very new and there's been industry funding that has generated that data, typically the industry partner needs to do their 6 months of those analysis and then we get access to that. So that's also something to consider. Indicate time points needed. So whether that be you want the longitudinal data or you just need a baseline measurement and subgroup requirements. So do you want the whole cohort? Do you only want those with AD? For the samples, obviously provide the type. So whether that's blood or CSF, as I guess CSF, as anyone who is versed in how CSF is collected through lumbar punctures, there's not that much CSF data available. So you've got to have a pretty strong justification for that. But that doesn't mean that that data is not, those samples are not shared. So as long as you provide the type, a really strong justification, volumes and assays used. And last thing is making sure that the aims in the project outline match the data and samples requested, I think is something that will end up requiring verification and things like that and back and forth. So if that's really clear, EOIs typically move through a little bit quicker. In terms of outcomes, stating whether the project is for pilot data, publications, developing methods, or potential IP is usually what you have to do. But it's all pretty straightforward, and Ying and I, or if any of you are at organisations where people have used AIBL data before, I'm sure they'll have their own tips and tricks. But I'm going to leave it there for our presenters, but I just wanted to say, obviously, we're presenting on the AIBL data, but the AIBL study is what it is because of the time that the AIBL participants have put into it over the years, and also all of the researchers and staffs who have contributed to the study. But yes, we'd love to see more people, particularly EMCRs, making use of the data set. So let us know if you have any questions or wanted to chat about project ideas. But yeah, we definitely encourage people to contribute.
Our next speaker is Dr. Eleanor O'Brien. Eleanor is a postdoc research fellow at the Edith Cowan University's Center for Precision Health and the School of Medical and Health Sciences. And today her talk is predicting age at onset of amyloid beta from genetic risk and resilience for Alzheimer's disease.
Okay. Thank you very much. Can you hear me? Yep. Good. Okay, so thank you for that introduction. I just wanted to start, before I go into the specifics of the study going to present, I just wanted to talk a little bit about the kind of research that I do, just a very broad overview of how I use AIBL data in my research. So I am a geneticist. I work in the genetics group in Centre for Precision Health at Edith with Tenniel. And my research is mostly focused on genetic factors associated with risk, onset, and progression of Alzheimer's disease. So I work a lot with the AIBL genetics dataset. But obviously, as geneticists, we can never work in isolation because the genetics is really only interesting to the extent that it can predict clinically meaningful outcomes. So we also collaborate really closely with all of the other streams who provide data on things like diagnosis, imaging, biomarkers, and cognition. And I also do a bit of work looking at interactions of genetics with lifestyle and environment. So we also collaborate a bit with the lifestyle group using data on things such as physical activity and sleep in particular. So as far as the techniques that we're using to link genetics with these different outcome measures, we do studies using targeted variants that may have been identified from previous studies, genome-wide association studies, or GWAS, where we're looking at variants across the whole genome, and also developing polygenic scores to predict particular outcomes. And the last two are things I'll cover in the talk I'm going to give today. So this is some work that we've been doing for a while, where we're looking at whether we can predict age at onset of amyloid beta from genetic risk and resilience for Alzheimer's disease. So just some background, as Tenniel's already flagged, we know that accumulation of brain amyloid begins a long time before cognitive symptoms, maybe decades before. And so if we've got a way to better predict the age at which brain amyloid is going to reach some critical threshold, then that maybe provides a window for administering treatment or other interventions to hopefully prevent or delay the onset of cognitive symptoms. And so we're interested in whether genetics can contribute to improving this prediction. And in the work I'm going to talk about today, this uses polygenic scores, which is, for any non-geneticists, basically a way of summing the effects of genetic variants across the genome to give us a single score that can help to predict someone's overall genetic predisposition for a disease or a trait. So the specific research questions that we're looking to answer with this, firstly, can a polygenic score developed based on genetic risk or resilience for Alzheimer's disease, based on previous studies of the genetics of risk and resilience, can this predict the age at onset of brain amyloid beta? And then asking whether we can improve on that prediction by constructing a polygenic score that's specific to this trait. Does that do a better job than just using genetic risk or resilience for Alzheimer's disease? So the population and data that we used for this was actually the ADPIC study, which brings together AIBL data with data from ADNI and OASIS, two North American longitudinal studies. So we had just over 1,100 individuals in total, and these people were all accumulators of amyloid beta, which we knew because we had longitudinal data, PET amyloid data for them. So that meant that they either had already had PET amyloid that was over 20 centiloids, or based on the longitudinal change over at least 2 time points, they had an increase in PET amyloid over time, and they were predicted to exceed 20 centiloids within 5 years. They all had genome-wide SNP data, that had been genotyped on a SNP array, and then to harmonise across the 3 cohorts, the SNPs were imputed against the TopMed imputation panel, and this gave us nearly 5 million SNPs that were common across all 3 cohorts. So we're defining here age at onset of amyloid as the age at which their brain amyloid was predicted to exceed 20 centiloids, and based on estimates from a natural history of PET amyloid that was developed in the reference you can see here. So the kind of workflow that we use to develop these polygenic scores, we started by extracting summary statistics from published genome-wide association studies for risk and resilience for Alzheimer's disease. And then we used these to find a kind of optimal threshold for which SNPs should be included to construct a polygenic score to predict our outcome variable. So basically what that means is we have, from the genome-wide association study, we have effect sizes and p-values for each of the SNPs in that, and the program we used, Precise, will run through a whole lot of different p-value thresholds to find the one that, if you use it to construct a score, will explain the most variation in the outcome, in this case, age at onset of amyloid. And when we did this, we excluded the ApoE region, which we know is going to explain the most variation in this trait. So what we're trying to do is construct a score that, that explains variation in this over and above the effect of ApoE. So we included ApoE as a covariate in the analysis, but it wasn't included in the score. So the score is looking at effects separate to effects of ApoE. And then, as I mentioned, we wanted to also look at whether we could improve on this by creating our own polygenic score that was specific to this trait. So we did that by separating our data into a discovery set and a validation set, running a GWAS in the discovery, and then using the same process to construct the polygenic score in the validation set. And because this is a relatively small data set for doing a genome-wide association study, just as to increase the stability of those, improve the stability of those measures, we did this 10 times. So we separated the data set into discovery and validation 10 times, and in each of those splits, we did the GWAS in the discovery and then constructed the polygenic score in the validation, and then also recalculated our risk and resilience polygenic scores in the validation to make sure we were comparing the same thing. So looking at the results, on the figure on the left shows the overall for the population, the relationship between our polygenic score for AD risk and then against the estimated age at onset of amyloid. And then the one on the right shows this separated by number of ApoE4 alleles. So as we would expect overall, someone who has a higher genetic risk of AD shows an earlier age at onset of amyloid. If we look at this separated by number of ApoE4 alleles, as expected, there's a really strong effect of ApoE, so you can see those lines are quite separate. So more ApoE4 alleles, there's an earlier age at onset, but also looking within those groups, just at the effect of our score, there was still this negative relationship between the score and the estimated age at onset within non-E4 carriers and E4 heterozygotes. So they're still showing, the score is still predicting an earlier age at onset even within those groups. And then looking at resilience, genetic resilience to AD, so a score based on this. Again, as we'd expect, people who are more genetically resilient to AD are showing a later age at onset overall. When we separate that by number of E4 alleles, this time we actually find that this score is a stronger predictor of age at onset in the E4 carriers. So with the risk polygenic score, it was the non-carriers. It's only marginal. It's not quite significant in the homozygotes, but you can see it's actually explaining, that's probably because there's not so many E4 homozygotes, but it is actually explaining more of the variation in that group than in the others. So if we look at what this means in terms of actual kind of numbers, in terms of age, the age that people are hitting this critical threshold. So this, just to begin with, is just showing the overall mean age at onset at the top overall, and then separated into each of the E4 groups, so non-carriers, heterozygotes, and homozygotes. But then if we look at what happens to these numbers if we pull them apart and separate out people who are in the top 20% for, in this case, AD risk, genetic risk of AD, and the bottom 20%. And so we can see sort of the effect of that, of being at those extremes on the age at onset. So overall, it made a difference of about 3 years. People who were at the highest genetic risk had age at onset about 3 years earlier than those who were at the lowest genetic risk. And it was also significant for E4 heterozygotes, where the difference was almost 4 years, but no difference in the E4 homozygotes, notice. And then the same thing, looking at genetic resilience to AD, wasn't quite so, the effect wasn't quite so strong, but there was still a difference. Overall, people who were most genetically resilient to AD had an age at onset that was about 2 years later than those who had the lowest level of genetic resilience. So, as I mentioned, we also tried to develop our own phenotype-specific polygenic score for this trait. And so this shows the figure on the left shows if you just focus on the coloured diamonds, so for risk, resilience, and then for our trait-specific score, we've got sort of an average across our 10 different cross-validation folds that we did, an average effect of that polygenic score on age at onset of amyloid. So you can see higher risk, it was significantly negative, higher resilience, significantly positive, but our phenotype-specific score didn't actually explain significant variation in this trait. So we're not improving on it over and above what you see from the polygenic scores based on risk or resilience to Alzheimer's disease. So in summary, people who are at higher genetic risk of Alzheimer's disease had earlier age at onset. This was still seen when we separated out by E4 carriage, with the association most significant in E4 non-carriers and heterozygotes. And we see 3 to 4 years difference in the age at onset of amyloid between the highest and lowest genetic risk scores, after accounting for effects of ApoE. So, and likewise, genetic resilience to AD, association was strongest in ApoE4 carriers. So more genetically resilient people had a later age at onset, and there was about 2 years difference between the highest and lowest resilience scores, but our phenotype-specific score was not associated with significant variation in age at onset. So in conclusion, in particular, genetic risk of AD was a significant predictor of age at onset of amyloid, and this was, importantly, over and above effects of ApoE. So ApoE obviously still is the strongest genetic risk factor, and that did explain a large proportion of the variation, but this score did still explain more variation over and above that. So if it's used in combination with other factors, it may be a useful tool that can improve prediction of people who may be at risk of early amyloid onset, and so provide a window for, or provide a way to identify people who may benefit from inclusion in clinical trials, or for early treatment or intervention to try and delay or prevent onset of AD. And I'd just like to acknowledge the participants and our funders and my colleagues and collaborators. Thank you.
Thank you, Eleanor. So our next speaker is Dr. Azadeh Feizpour, and Azadeh is a senior research fellow at the Department of Molecular Imaging and Therapy, Austin Health. Today, her talk is From Data to Diagnosis, How AIBL and ADNeT Advance Alzheimer's Research.
Okay, so thanks to Neil and Ying for organizing this webinar. In this presentation, I'll, as Ying mentioned, talk about how we leverage AIBL and other datasets like ADNeT to address important questions from different sectors in terms of Alzheimer's disease research, focusing on blood and imaging modalities. So I thought to start from this presentation of the last advancements in this field in the last 25 years, that going back to before 2000, that the only way of diagnosing Alzheimer's disease for certain was post-mortem examination of the brain, to today that we use blood biomarkers for detecting amyloid and tau pathology in the blood. And in between that, it was only 1999 that the first promising amyloid PET tracers came out, and 2013 the first promising tau PET tracers came out. And the reason I'm mentioning this is to highlight the importance of cohort studies like AIBL, because AIBL encompasses the most important data that we can have for addressing important questions using a lot of amyloid PET, tau PET, and blood biomarker data that we've accumulated in the last 20 years. The questions that we address using AIBL come from different AIBL dataset, come from different sectors. This could be from pharmaceutical companies that, especially in the peak of blood biomarker discoveries, every couple of months we had a new blood test that was coming out, and these pharma companies want to validate this blood test, and AIBL provides that opportunity. CROs want to know whether they can use blood tests for screening purposes. Clinicians want to know if they can use it in clinics for diagnosis and prognosis in a more affordable way. And patients and participants hear about these discoveries in news and want to know how that applies to them. So I'm going to address three of those in the interest of time. The first one being whether these new blood tests that are coming out are accurate reflection of amyloid and tau. And in AIBL, as I said, we have the opportunity to have a lot of blood samples that we send to companies for testing testing level of different assays. For example, plasma p-tau217 from Fuji Bio, that we have the matching amyloid and tau PET for these individuals. And to validate such novel blood tests, we do some correlational studies. For example, in this instance, on the y-axis, we have the novel blood test, which is the Fuji Bio p-tau217, and plotted against amyloid PET quantified in centiloid scale. For two tests, one novel, one gold standard, that had positive correlation, you would want majority of the data points to be in the bottom left and top right quadrants. No test is going to be perfect, so we will always have false positives and false negatives in the other quadrants as well, but this is already looking very good. When you look at this, you see a very good test. But we usually like to quantify things, and that's using ROC AUC. So in this instance, to assess the performance of p-tau217 from Fuji Bio against for discriminating amyloid PET status, we plot the sensitivity and specificity of this novel test, and we look at the area under the orange curve, which is the AUC or area on the curve. And in this instance, it is 93%, the closer to 1 the best, 100% the best, the better. And in this case, 0.93 is a very good performance. But AUCs could be misleading. We also, for validating this test, look at PPV and NPV, positive and negative predictive value, which in this case, the positive predictive value is 93%, NPV not that good. And this is mainly because of the high prevalence of disease in this cohort that we looked at. That's why we look at different scenarios with different prevalences for validation purposes as well. And this can be repeated with tau PET. So in this instance, we've looked at performance of p-tau217 against tau PET, and the area under curve was 0.94. Again, very good performance. But our research in this area is not limited to diagnostic accuracy. The other questions posed to us is, for example, prognostic performance of these blood tests. And we address them by looking at the prognostic performance of the blood biomarkers, such as, in this case, again, p-tau217 from Janssen, comparing to tau PET and amyloid PET. In this instance, on the y-axis, you have CDR sum of boxes, an indication of cognitive performance. And the curves look at the binarized value of each of the biomarkers. And again, we like quantifying things. So we've looked at the beta coefficient from regression analysis to compare the performance of these tests to each other. And as you can see, p-tau217 as performance in predicting changes in CDR sum of boxes is very close to tau PET. And that's a very promising performance for a blood biomarker for prognostic purposes. The other area I wanted to tap into was the use of this new blood test for clinical trial screening. So a couple of years ago, the revised criteria for diagnosing and staging of Alzheimer's disease came out, and based on that, we include all of the amyloid positive participants, but these 4 stages apply to people based on their tau in the brain, and that's the location of tau and the amount of tau in the brain. So if somebody doesn't have tau, that's a stage A. If they have tau limited to medial temporal brain region, that's a stage B. If they have it in neocortical region in moderate level, that's stage C. And high level of neocortical tau, that's stage D. The reason I'm mentioning this is that for clinical trials, it's important to discriminate these stages, because in the last a few anti-amyloid treatment trials, we've seen that people with high tau do not respond to these treatments very well. So what we also, again, leveraged our AIBL data set to address was whether we can use these blood biomarkers to be able to discriminate between these stages. So first of all, this is a plot of these 4 stages in addition to A negative, T negative for comparison against Fuji bioplasma p-tau217. And we put this to test to see whether this blood biomarker can exclude people that have high level of tau. And for that, the area under curve was 0.89. But as you can see, the NPV is pretty good, which means we meet what we wanted. The people below the threshold indicated here, 95% of them would actually be within the groups that we want with intermediate and low level of tau. But the pretty low PPV show that we're also excluding a lot of people that could benefit from this test. So we're still looking into this using different biomarkers. And again, AIBL provides this opportunity for us to send the blood samples to pharmaceutical companies that have more promising blood biomarkers or combination of them to be able to discriminate this better. Going back to this slide, these were a few examples of many research that we're doing in this area by different people, and also implementing this blood test in memory clinics to be able to maximize the benefit of this in terms of affordability and accessibility for the community. So if any of the presentations today pop up any questions, research questions that could be addressable by AIBL dataset, feel free to reach out to us. These are my acknowledgements, and thank you.
Thank you, Azadeh. So our next speaker is Dr. Emily Rosenich. Emily is an Alzheimer's Association Research Fellow at the Turner Institute for Brain and Mental Health within the School of Psychological Sciences at Monash University. So her talk is Differential Effects of ApoE and the Modifiable Risk Factors on Hippocampal Volume Loss and Memory Decline in A beta negative and A beta positive older adults.
Thank you so much, Ying. So thanks for the invitation to speak to you today. So for those of you who don't know me, I'm Emily. I'm currently working at Monash University as a postdoc within the lab of Professor Yanying Lim. And I'm just going to get straight into it. So many of you here today will be familiar with this figure. This is a seminal figure from a Lancet commission that was updated most recently in 2024 by Jill Livingston and colleagues. And this study identified 14 modifiable risk factors for the development of dementia. And together, these risk factors are proposed to account for up to 45% of all-cause dementia cases. And interestingly, 7 out of these 14 modifiable risk factors are actually vascular in nature. Unfortunately, though, not all risk factors for dementia, as we've learned throughout these presentations, are actually modifiable. So there have been other non-modifiable risk factors identified for the development of cognitive decline and Alzheimer's disease. These are older age, being of the female sex, as well as genetics. So as it was talked about previously, carriage of 2 ApoE4 alleles is the strongest genetic risk factor for the development of Alzheimer's disease, and ApoE is thought to increase risk for Alzheimer's through increased amyloid accumulation, but also through other non-amyloid related pathways. So for example, its role as a lipid or cholesterol transporter, as well as its genetic risk factor for the development of both cardio and cerebrovascular disease. So in light of the fact that there appears to be multiple modifiable as well as non-modifiable risk factors for the development of dementia, I'm going to be particularly focusing on Alzheimer's dementia today. There's been these dementia risk scores developed, which, as the name suggests, attempt to predict someone's future risk of developing Alzheimer's disease. And one example of these risk scores is a risk score called the Cardiovascular Risk Factors Aging and Dementia Risk Score. And that risk score includes factors like age, sex, ApoE, education, but also vascular risk factors like cholesterol, BMI, physical activity in its computation. And these risk factors are really important because, these risk scores are really important because we need them to be able to identify people who are at the highest risk of disease, so that these people can be identified and stratified appropriately into clinical trials focused on primary prevention. So these risk scores often show higher clinical utility as well, compared to using just individual risk factors on their own, particularly when we're talking about vascular risk factors, because they're actually known to cluster together or co-occur. However, the extent to which both these modifiable and non-modifiable risk factors contribute to the development of cognitive decline and brain volume loss, so neurodegeneration, in older people with different brain amyloid status remained unclear. So that's why we wanted to utilise the amazing data within AIBL. Our aim of this study was to determine the differential contributions of both those modifiable and non-modifiable KD components, with episodic memory decline as well as hippocampal volume loss in people who were cognitively normal, for whom we had brain amyloid status available. So our age range between 58 and 91, participants had to complete at least 2 neuropsychological assessments, as we were interested in looking at this longitudinally, and they had to have completed an amyloid PET scan so that we could determine whether they were classified as amyloid negative or amyloid positive. So in terms of our computation of our risk scores, for each person, we computed both the regular KD risk score, which included all of the variables in the left-hand side of that table, as well as a purely modifiable version of this risk score using only the variables highlighted in green text in that table. So for our stats, we ran a series of linear mixed effects models, including 3- and 2-way interactions. So for example, between amyloid status, our risk scores, and time on hippocampal volume and episodic memory. And because we wanted to assess those differential associations, we looked at all the non-modifiable variables from the KD. So age, sex, and E4 were included as separate predictors in all of our modifiable KD models. So in terms of what our sample looked like at baseline, most participants were female and the mean age was just shy of 71 years. Most participants had an education level of about 12.5 years on average. And compared to participants who were amyloid negative, amyloid positive participants at baseline showed lower cognitive performance, lower hippocampal volumes, and they also showed higher KD risk scores, and they had a higher proportion of E4 carriers as well. So in terms of our main results, we did find a significant amyloid group by KD by time interaction for hippocampal loss. And that effect, interestingly, was actually being driven by participants that were amyloid positive. So such that in that group, having a higher KD risk score, so being at greater dementia risk, was negatively associated with having a greater rate of hippocampal atrophy over time. And interestingly, we didn't find any significant 3-way interaction for episodic memory decline here, suggesting that those 2 amyloid groups were showing equivalent rates of decline on the episodic memory test. However, something really interesting happened when we pulled apart the components within the CATI. So we actually found the opposite thing here. So we found no significant amyloid group by modifiable CATI by time interaction for hippocampal volume loss, but we did find a significant three-way interaction for the modifiable CATI. And this was really being driven by those amyloid negative participants, such that those participants, they had a higher modifiable CATI risk score, and that was negatively associated with them having a greater rate of episodic memory decline over time. And when we pulled this apart in exploratory sensitivity analyses, when we put all the individual modifiable risk factors in the model as separate predictors and controlled for the non-modifiable risk factors, we found that it was really higher BMI that was driving this effect. We also looked at amyloid group by ApoE4 status by time interactions, and we found that compared to participants who were E4 non-carriers, E4 carriers, irrespective of amyloid grouping, showed greater hippocampal volume loss and greater episodic memory decline over time as well. So what does this all mean? Well, firstly, aligning with previous studies, we found that ApoE4 is associated with an increased rate of episodic memory decline and hippocampal volume loss in people who are classified as amyloid positive and are cognitively normal, but we also extended that in this study to show that that relationship exists in people who are amyloid negative as well. We've also found that in amyloid negative cognitively normal adults, having a lower prevalence of modifiable risk factors that were mostly cardiovascular in nature was associated with less episodic memory decline over a period spanning about 10 years. And that finding is really important and interesting because it suggests that interventions to prevent or delay cognitive decline might actually be most useful in people who are at high genetic risk of Alzheimer's disease, but who are not yet classified as amyloid positive. So really emphasizing the message that early intervention, particularly with lifestyle intervention, is best targeted to those who are earlier in life. So for example, in midlife stages. So with that, I would just like to acknowledge the organisers of this presentation today, the ADNeT and AIBL teams, all of my collaborators on this project who are listed on the left of this slide, and of course, all of the AIBL participants, because none of this research would be possible without their dedication and commitment. So thank you very much.
Thank you. So I guess I will let all the speakers unmute themselves, and then now we open to the questions.
Also, you can either type the questions into the Q&A box, or if you raise your hand, then you will be able to allow you to speak and unmute. Lizanne?
Hi.
Hello.
Yes. I was wondering if there's any data regarding menopause in this AIBL data set.
That is a great question. That's a great question. There is hormone data. So there's hormone data and there is medication data, but I am unsure off the top of my head unless anyone else knows.
I remember this coming up in a meeting. I mean, most AIBL participants are over 60.
Yeah, they would be over 60, 65, so you would expect. Yeah.
Okay, thank you.
But I guess, I don't think there's data on the age that people went through menopause, which could be something that.
Yeah,
that would be interesting. But yeah, there's hormone data and there is medication data. So I guess that might tell you a little bit about the point and where obviously things change.
Thanks.
No worries.
I think that taps into Edwin's question in the Q&A.
Yeah. Yes, so prescription medication is recorded in AIBL. So there's recently been a large project that's been underway because essentially it was a lot of self-reported medication and they've gone through and essentially classified. So there are large classifications going to more specific classifications around the type of medication that our participants are on, because as you can imagine, it's quite wide-ranging. I guess to any of the speaking for the current projects that you're doing, or the projects that you've presented on, if there was anything additional that you would have liked to be included, is there something that, or is there something that you feel like, or is there something that's now within AIBL that you feel like you could have incorporated that would strengthen, or something that you would add in if you were to do the analyses again?
I can go first. I guess one of the things that we haven't, at least to my knowledge, we haven't looked at in AIBL is effective. Other research groups across Europe and the US have looked at effective medication on blood biomarkers.
Yeah.
And I think we haven't gotten yet to that stage. I mean, you mentioned that now we've classified them to larger classes of medication, which is very helpful, because I always dreaded looking into medications because each person put in like 10, 20
Yeah.
that age. So yeah, so if there are larger classification, broader classifications, that now enables us to actually go in and have a look at the effect of those on blood biomarkers and whether that moderates the correlations that we've reported before. Yeah, that'd be interesting.
Yeah. There's another quick question in the chat. So cardiovascular risk factors. So in terms of the data that we had, we do have medical history. So we've got something that gets used quite a bit is a score of cardiovascular disease risk. So that's a combination of medical history of hypertension, stroke,
diabetes,
and
the BMI.
Yeah, so there's BMI as well. We definitely have medical history of diabetes. And then within those, we also have a little bit of information on, again, some biomarkers. So things like insulin level, glucose level, measures of insulin resistance, and then we have measures of LDL, HDL. And that's
I wanted to add to that as well, because that was something that was really nice about working with the AIBL dataset, is that in a lot of large datasets, cardiovascular risk factors are self-reported and they're often categorized as binary variables. So essentially diagnosis versus no diagnosis or past diagnosis. Whereas AIBL actually has more objectively measures of these cardiovascular risk factors. So blood pressure readings, for example, levels of total cholesterol, which I think is a real strength of this data set compared to a lot of other data sets which just have self-report diagnosis, yes, no.
I think it's 11 o'clock, so I think all of us are pretty happy. If anyone has any questions that they think of after this, if people want to email any of those through, happy to answer any questions that people have. Everyone.
Thank you. Have a good afternoon.
Thank you. Bye.
Bye.